To understand biological data storage, we must first look at the physical limits of the cell. Take the scale of a single bacterium. This is an electron micrograph of E. coli. It represents the baseline scale for autonomous life. Inside this microscopic rod, the organism stores its complete genetic operating system, a sequence of exactly 4.6 million base pairs of DNA. A human diploid cell scales that requirement up drastically. The human nucleus holds roughly 6.6 billion base pairs. In a DNA molecule, the physical distance between any two sequential base pairs is a rigid biological constant, 0.34 nanometers. If we multiply those 6.6 billion human base pairs by the 0.34 nanometer distance, the result is exactly 2.2 meters of linear DNA. That 2.2 meter thread must operate inside a cellular nucleus measuring roughly 6 micrometers across. Achieving a compression ratio of hundreds of thousands to 1 while maintaining active read and write access to the data is an extreme physical engineering problem. We can only map how the cell achieves this by deconstructing the atomic architecture of DNA from the ground up. The fundamental building block of this genetic vector is the deoxyribonucleotide monomer. It is built from 3 distinct chemical components: a pentose sugar, a nitrogenous base, and a phosphate group. The central piece is a 5-carbon pentose sugar, identified specifically as deoxyribose. Compared to the sugar found in RNA, it lacks a hydroxyl group, an oxygen atom, at its 2' carbon. The first linkage occurs when the nitrogenous base attaches to the sugar's 1' carbon, secured by an N-glycosidic bond. The second linkage snaps the phosphate group onto the sugar's 5' carbon via a phosphoester bond. To build a sequence, these isolated monomers must polymerize. The 5' phosphate of one nucleotide binds to the 3' hydroxyl of the next. This creates a continuous 3' to 5' phosphodiester bond. For any linear, double-stranded sequence, the mathematical stoichiometry dictates that the total number of these phosphodiester bonds equals the total number of nucleotides, minus 2. This asymmetrical alternating sequence of sugar and phosphate forms a rigid directional backbone. It serves as the physical prerequisite for stable, long-term data storage. To complete the structure, a second polynucleotide strand must align with the first. Because of the backbone's chemical asymmetry, this second strand must run anti-parallel, aligned upside down from the 3' to the 5' direction. The interior connections are governed by Chargaff's rule. This chemical law requires that large double-ring structures, the purines, strictly pair with smaller single-ring structures, the pyrimidines. Specifically, the purine adenine pairs with the pyrimidine thymine, locking together using exactly 2 hydrogen bonds. Guanine pairs with cytosine, using exactly 3 hydrogen bonds. The geometric imperative of Chargaff's rule is strict spatial uniformity. Pairing a double ring with a single ring ensures the interior rungs are always exactly 3 rings wide, locking the overall helix width at precisely 20 angstroms. Macroscopically, this linear ladder structure naturally twists upward into a right-handed helix. The pitch of this helix is mathematically locked. It takes exactly 3.4 nanometers to complete a full 360-degree turn, which encompasses exactly 10 base pairs. Strict geometric uniformity, a continuous 20 angstrom width, and a fixed 3.4 nanometer pitch yields a predictable physical structure that can be reliably stacked, coiled, and compressed. The primary chemical hurdle to packing this 2.2 meter genome is the backbone itself. Because the continuous chain of phosphate groups carries a dense negative charge, if forced into close proximity, the strands violently repel themselves. Biology overcomes this repulsion by deploying a class of proteins known as histones. These proteins are densely populated with positively charged basic amino acids like lysine and arginine. 8 of these histone proteins aggregate to form a massive cohesive cylinder called an octamer, which projects a strong positive electrostatic field. The negatively charged DNA strand is intensely drawn to this positive core. It wraps around the octamer, securing approximately 200 base pairs of DNA. This spooling creates the foundational packaging unit, the nucleosome. This creates a higher-order topology. Multiple nucleosomes repeating in series form a structure resembling beads on a string, which then loops and coils onto itself to generate thick chromatin fibers. Through this hierarchy of extreme compression, the loose chromatin condenses into the final, recognizable form of a metaphase chromosome. The eukaryotic genome is packed through a precise sequence of electrostatic engineering. By neutralizing the DNA's natural charge, the cell solves the 2.2-meter spatial paradox. Knowing this physical structure allows us to revisit a critical historical dilemma. In the mid-20th century, scientists were unsure if genetic data was carried by the DNA molecule or by the complex proteins packaged inside the nucleus. Initially, proteins were the favored candidate. Their vocabulary of 20 distinct amino acids seemed mathematically capable of far more data complexity than DNA's simple 4-base alphabet. To resolve this debate, researchers turned to a highly specialized biological tool, the bacteriophage. A bacteriophage has an elegant binary composition. It is built entirely of just two components, an exterior protein coat and an interior core composed exclusively of DNA. This provided a distinct biochemical separation. Based on the structural architecture we've mapped, DNA possesses a dense phosphorus backbone but entirely lacks sulfur. Conversely, the protein coat possesses sulfur but entirely lacks phosphorus. In 1952, researchers Alfred Hershey and Martha Chase designed an isotopic tracking strategy. They grew one batch of phages in radioactive phosphorus-32 to selectively light up the DNA, and a separate batch in radioactive sulfur-35 to selectively tag the protein coat. This isotopic isolation created a non-overlapping tracking system. Whichever tag crossed the cellular boundary and entered the bacteria would definitively identify the molecule of inheritance. The experiment began by allowing tagged bacteriophages to infect the E. coli. Next, mechanical agitation sheared the viral husks off the bacteria. Finally, a centrifuge forced the heavier cells down to form a pellet, while lighter husks remained suspended. In the sulfur-35 batch, the radioactive tag was found exclusively in the lighter supernatant. The protein coat never entered the cells. In the phosphorus-32 batch, the radioactive signature was found deeply embedded inside the heavy bacterial pellet. The physical data yielded a definitive conclusion. The viral DNA, and only the DNA, had crossed the cellular boundary to hijack the host's machinery. This proof relied entirely on the molecular architecture of the nucleotide. It was the specific existence of continuous phosphodiester bonds and the phosphorus contained within them that allowed researchers to track the molecule's movement. The unique geometry of the DNA backbone solved the physical paradox of genomic compression. Simultaneously, its distinct biochemical signature allowed researchers to track its movement, isolate its location, and definitively identify it as the true source code of life.